Hello everyone, this is Monica-san from GenScript. In this webinar, I am going to talk about the current methodologies and bottlenecks of Western blotting, and more importantly, how to accelerate Western blotting with the optimal solutions while maintaining industry standard quality. There are various research techniques in the field of protein analysis, and Western blotting is one of the most useful tools for protein detection and analysis. It has been widely applied in many research topics, such as biochemistry and proteomics, because of its gold standard quality and essential role in immunodetection. However, a couple of challenges faced by many researchers out there in the field include its non-skippable sequential procedure, from loading and running samples, transferring to membranes, blocking, antibody incubation, and multiple washes. This is the universal procedure, which takes hours to days, that is labor-consuming and time inefficient. And second, its inaccuracies and inconsistency associated with all the manual procedure, which causes low reproducibility in data and potential waste of samples. With that being said, how can we dissect the challenges and resolve them in each step is the project we all try to work on. Western blotting normally includes these lab procedures: gel electrophoresis, gel transfer, blocking and incubation, and sometimes gel staining. I will go into details of each step to address the challenges or common issues and how we can select the optimal solutions to achieve better and more consistent results. Tools or lab consumables related to gel electrophoresis is apparently the gels, both precast or homemade gels. This is the step when denatured proteins are loaded into gels and run an electrophoresis to separate the protein bands by molecular weight. The common issues in the protein separation step usually is the smiley or smearing bands, or sometimes the homemade gels are too fragile to handle with. Gel selection can be optimized from several technical perspectives, including the types and concentrations of polyacrylamide, which is the common composition of precast gels, and sample preparation reagents. First of all, by comparing to homemade gels, precast gels have a more uniform manufacturing process, including control on temperature, oxygen level, chemical purity and freshness, and consistency of the casting profile. So I'll focus on talking about selecting precast gels in this webinar. So what are the factors to determine high-quality gel electrophoresis using the precast gels? The precast gels are mainly polyacrylamide gels. They are polymerized by a mixture of acrylamide, which are the long polymers, and bisacrylamide, which are the short cross-linkers. When these two types of polymers react, they form into this porous gel. The pore size is determined by the concentration of both acrylamide and bisacrylamide. Also, there are other factors such as the initiator and catalyst in this polymerization reaction. But the most important or influencing factor here is the concentration of acrylamide. Different brands have different concentrations, which result in different separation performance as well. You want the proteins to migrate in relatively fast speed in a larger pore size gel, but you also want stability and band consistency. So it's important to have it balanced, and that's how GenScript gels generate premium separation results and a fast run based on both of that. The second factor is the sample loading buffer. Before loading the protein samples, we usually heat samples with SDS and DTT to denature the proteins. And pH of the common sample loading buffer will decrease from 6.5 to about 4.5 upon heating, which could not completely break the disulfide bonds in most proteins. GenScript has a sample loading buffer that its pH can still remain at 7 and thus helps with better protein denaturation and separation. Another two factors are both benefits of the bistress gels. and it has higher operating pH while running to increase modification of the proteins, and neutral casting pH to avoid hydrolysis, and thus is more stable for storage and has a longer shelf life. With those technical information being shared, I would like to introduce the short-page precast bistress gels developed by GenScript to separate a wide range of protein sizes by electrophoresis. It can generate sharp and consistent bands, which is the most important, fast run times while pertaining the good separation quality, room temperature storage supported by its bistress composition, so it's quite stable at room temperature. Running buffer included in each pack, and large loading volume because of our special well design. As a precast gel, you can see how it generates the band consistency here, high sensitivity, and clear and flat bands that are not bent at the edge. To run gel electrophoresis, there are some other consumables involved. For optimal and consistent results, we also recommend using the full set of solutions to make the process easier for you. Gel tank is the most essential tool, which supports for up to 2 mini gels. It's also compatible with our traditional blot module for transfer. Second, the 4X LDS simple buffer is the one I mentioned that has a higher pH for more complete protein denaturation. MES and MOX running buffer is the buffer you pour into the gel tank for fast run. These two options can fulfill a wide range of protein sizes. Last but not least, the protein ladders. These are the sharp and precise markers for protein electrophoresis. The pre-stain we have is the most popular one, which ranges from 5 kilodaltons to 270 kilodaltons. And there's also unstained ladder and Western blot viewable ladder. Both help as a troubleshooting reference along the Western blotting procedure. So after gel electrophoresis, gel transfer is the second procedure in Western blotting. Related tools are automated transfer systems or traditional wet transfer, the blot module. Most common issue in this step is the difficulty in transferring large proteins. In order to find the optimal solution for this gel transfer procedure, let's first take a look at the comparison chart between gold standard wet transfer and automated fast transfer device. In this gel transfer technology comparison slide, I will compare the three methodologies, which are the traditional manual wet transfer blot module, semi-dry or dry automated transfer device, and automated wet transfer device. Comparison will be made from 5 aspects: transfer time, protein size coverage, transfer efficiency compared to the industry standard, its benefits and limitations. So first, transfer time. The traditional wet transfer blot module takes about 1 hour to overnight. The semi-dry device on the market usually takes about 9 to 15 minutes, and the automated wet transfer device we have takes about 9 to 17 minutes. The automated instruments definitely save more time compared to the traditional one. The traditional one, because of its gold standard wet transfer, it can technically transfer full range of proteins. The larger the protein is, the longer transfer time it takes. The semi-dry device, even though it's very fast and convenient, the size coverage only ranges from about 3 kilodaltons to 200 kilodaltons, and it's usually challenging to transfer larger proteins. The automated wet transfer instrument, because it incorporates the wet transfer technology, it has a wider protein size coverage from 3 kilodaltons to more than 1,000 kilodaltons, giving more benefits to the researchers to transfer large proteins. In terms of transfer efficiency, since we are comparing to the traditional methodology as the gold standard, semi-dry is good for smaller proteins only, and automated wet transfer has comparatively good efficiency compared to the industry standard. benefits of the traditional one is that it's reliable and cost efficient, but the manual operation is messy and labor consuming and sometimes has bubbles hidden between the gels and membranes, which reduce the transfer performance. The semi-dry or dry devices on the market, they are fast and convenient for sure, has a clean setup, generates consistent results. But limitations are that it's an inefficiency to transfer large proteins because it's not wet transfer technology, and its lack of buffer to support the larger protein transfer. And also high cost on consumables is another concern of many lab researchers. For the automated wet transfer device, it has the benefits of automation and fast. It has pre-packed reagent kit to ensure convenience and consistency. The limitation, however, is that it requires more bench space than the blot module and semi-dry devices. Going back to the original question, how do we find the optimal solution from the market for gel transfer? Now we know that we have the traditional manual wet transfer, which has the gold standard transfer efficiency, and semi-dry or dry transfer device that is fast and convenient in operation, guaranteed result consistency, but they each have their own limitations. GenScript combines the benefits of these two common methods while eliminating the limitations to invent eBlot L1, the automated wet transfer system. It has the high reproducibility and efficiency of traditional wet transfer with the speed and convenience of semi-dry transfer systems. Its unique and clean transfer sandwich setup design eliminates the messy and redundant preparation and cleaning processes. This is also easy to assemble using the dry sponges, gel, membrane, cassette according to the instructions. eBlot L1 Fast Wet Transfer System has simple and user-friendly design, including its user-friendly interface, which supports simple hands-on operation. eBlot can run 2 gels in separate chambers, individually, and even with different transfer protocols that fulfill your need for a wide range of protein sizes. The display shows separate running status and times for each channel. The keypad supports easy operation, and buffer connecting parts on the side allow for clean buffer and water to be used for each cycle, and automated drainage of waste solution. The second feature is the automated fast transfer. The screenshot here shows only 17 minutes for a default standard protocol for common protein sizes. For smaller than 30 kilodaltons, we also have short protocol, which only takes about 9 minutes. And for larger than 300 kilodalton proteins, we have a long protocol, which takes about 23 to 24 minutes. The entire process is automated and fast. And next, we have the all-inclusive consumable kit. These are pre-packed transfer kits compatible with eBlot instrument. The kit includes pre-cut nitrocellulose or PVDF membranes and concentrated transfer buffer. Everything is prepared in the box for you to start using the eBlot device. In addition to its unique design and convenience, eBlot also maintains high transfer efficiency for all size proteins. Comparing to the traditional wet transfer and semi-dry instruments, eBlot has a higher transfer efficiency for both small proteins on the bottom row here and larger proteins on the top row here. Data on the right side also shows a demonstration of using eBlot to transfer up to 1,200 kilodaltons protein in 24 minutes, which is the long protocol on eBlot. Since eBlot keeps the gold standard wet transfer technology and its automated design, that's how it's able to support full protein size coverage. After gel transfer, I think this next step, blocking, incubation, and washing, is the step which gives most people headaches. It's the most tedious and repetitive procedure in Western blotting. Inconsistent results are sometimes inevitable as well. And available tools on the market are either the traditional shaker and manual operation or automated membrane processing system. Here you can see the common procedure of the Western blotting workflow, from blocking, primary antibody incubation, 3 times washing, secondary antibody incubation, and another 3 times washing. There's a lot back and forth manual operation in this process and takes hours to 2 days to finish the whole process. If we replace all the manual work with a fully automated membrane processing device, it can free you from a super time and labor consuming process. eZwest Lite is a benchtop friendly device about the same size as a shaker that can help you automate all these steps on the left. It's completely hands-off, generate consistent results, it's programmable, and also antibody recoverable. eZwest Lite is a fully automated Western blotting device. It has a user-friendly touchscreen that allows protocol customization. So any special or unique protocol you have developed for your samples or antibodies, you can input and save them in the program. You can also change and optimize as needed. We have the removable membrane cassette, which is for membrane loading. So after gel transfer, when you have the protein bands transferred to the membrane already, place the membrane inside the cassette for blocking and incubation in this device. Since the cassette is removable, you can also incubate it at 4 degrees for overnight. In the eZwest program, there's also an overnight option for you to select. One membrane cassette can incubate 2 membranes at a time. As I said, eZwest can do all the blocking, incubation, and washing steps in one operation. And here's where you load the reagents for one time. Antibodies are also recoverable back to the original reagent tubes here. Different from eBlot that only our pre-packed consumable kits are compatible and can be used. For eZwest, you can use your own reagent or solutions, so you can prepare your own solutions to be used to start using this instrument. Last but not least, we also have a compatible eZwest diluent kit, which is a fast blocking reagent. If you would like a more speedy process, this fast blocking reagent shortens incubation time and also skips the blocking step as well. The combination of the eZwest diluent kit and the eZwest automated device, the combination is definitely more time efficient and convenient. Here's the data demonstration comparing traditional milk blocking reagent on a shaker versus eZwest with diluent kit combination. As you can see the time used in each step, adding up to 4 hours for the traditional method and only about 1 hour and 50 minutes for eZwest. eZwest also generates, if not better, but similar results to the traditional way, so its automated design is not compromising the gold standard quality for our researchers. In addition to the common Western blotting procedures, some biochemistry labs also do a lot of gel staining. There are many staining reagents out there, but still messy operation and time-consuming are the common issues for this procedure. There's also one automated staining system, and I will go into the detailed comparison on the next slide. In this section, I will talk about the comparisons among stain-free system, silver staining, fast coomassie staining reagent, and automated coomassie staining device from 5 aspects as well. I will briefly go over the technical description of each one very fast. In the stain-free system, proteins are made fluorescent and can be immediately visualized in a gel containing a trihalo compound. So the time needed for visualization is very fast and immediate. Its benefits include the strong protein signals with low background and immediate visualization, while the limitations are that it's only able to react with tryptophan in proteins. and only able to visualize the trihalo compound as well. And it's also not compatible with the regular page gels, so the tools needed for this method is stain-free gel and imager. Second, the silver staining is when silver ions interact and bind to the proteins and deposit into the surface of gel at the location of protein bands, which requires about hours to overnight to visualize. Its greatest advantage is that it has nanogram level sensitivity while it involves multiple manual steps and very time consuming. It requires silver staining reagent for this method. The third method, which I think is also the most common method, is the fast coomassie staining reagent. It's the gold standard coomassie blue staining of gels to visualize protein bands, which takes about 20 minutes to overnight depending on the sensitivity. It is gold standard technique and very cost efficient, but messy, time consuming, and inconsistent results sometimes. The consumable needed is only the coomassie blue staining reagent. Last is the automated coomassie staining device developed by GenScript. It's an automated system integrates the traditional 3 steps of fixing, staining, and destaining into one to simplify the process. Page gels can be stained and destained within 10 minutes using coomassie blue reagent using this device. Its biggest advantage is its automation, fast, easy, and clean setup, and consistent results. Limitation is that it requires more benchtop space compared to the other 3 methods. Tools needed are the automated device and coomassie blue reagent. By looking at this comparison table, it's easier to see that the best substitute of the gold standard staining method is the automated device, when it has better staining results in a much faster time. It also doesn't limit to visualize the proteins only based on reaction with the tryptophan bands. So based on that, I would like to introduce more about our automated coomassie staining device. It's called eStain. eStain L1 fast staining system is the only automated coomassie blue staining device on the market. Similar to eBlot transfer system, it can stain 2 gels at the same time and also individually in separate chambers. It's fully automated and fast, only takes about 10 minutes to complete the full staining and destaining process. It has the sandwich setup that is easy to use and can be inserted into the chamber. It's clean and convenient with pre-made coomassie blue staining solution. In addition to its automation, fast, and convenience, eStain also generates consistent results with the blue gel staining with high sensitivity as low as 12.5 nanograms. For researchers that do membrane staining, eStain can also support membrane staining function with the Ponsus S solution. Here's more data showing comparison between eStain results and other common methods. eStain generates better sensitivity than both traditional manual staining and stain-free system. This is a brief showcase of how easy to use and clean the eStain setup is. The first step is to place a gel on the eStain gel holder, which is compatible to all kinds of mini gels. Second, cover the gel with a clean and wet filter paper provided in the pre-made package and insert the gel holder into the chamber. And then start the automated program for 10 minutes, staining and destaining. And after 10 minutes, take out the gel holder and check results. This entire process doesn't involve any hands-on touch to the staining solution and make sure your lab environment is clean, and all of these can be completed in 10 minutes. To recap what I included in this webinar, we've always been struggling with the traditional method, traditional Western blotting that takes up to 2 days, but GenScript solution allows the full process to be completed in as short as 2 hours with all the optimization. We keep the industry standard technology with reliable results, keep inventing automated solutions to save labor and time, and generate consistent quality. If you find any of these content interesting or would like to review the webinar again, please go to our website to learn more or download the comprehensive brochure. On the website, we also have the demo videos of instruments for you to check out. If you have any questions, please feel free to contact us through the email here or go online to request for quote. We will be happy to assist you and your research in the near future. Thank you.